Hello students, this is Molecular Basis of Inheritance part 6. In molecular biology, DNA replication is the biological process of producing two identical replicas or two identical or similar, exactly similar copies of DNA from one original DNA molecule, that is one parent molecule. uh, DNA replication occurs in all living organisms, acting as the most essential part of the biological inheritance. And the DNA replication occurs in few steps. Out of these few steps, we are going to study each and every step in detail, which is given in your syllabus or in your textbook. Out of these many steps, first step is activation of nucleotides. So if you're able to observe this diagram, these are the nucleotides which are activated. Okay, so there are four types of nucleotides of the DNA, that is DAMP. A stands for adenosine monophosphate, GMP guanosine monophosphate, CMP is cytosine monophosphate, and thiamine monophosphate. They are present in the nucleoplasm. Now what is nucleoplasm? That again you have to understand very clearly, that nucleoplasm is the substance of a cell nucleus. So nucleoplasm is the substance which is present in the nucleus, especially that not forming the part of a nucleolus. So in that nucleoplasm, these many nucleotides of the DNA, they are present, or they are suspended. So they are activated by ATP in presence of an enzyme that is called as phosphorylase. Now see, whenever the if you are studying DNA replication part, it is very much essential that please memorize, please try to understand which step you are studying, and in that particular step which are the enzymes. The name of the enzyme is very much important if you want to score good in the DNA replication, and it's very easy. The name of the enzyme, it functions. Okay, the name of enzyme, there is connection between the function of an enzyme and the name of the enzyme. Just try to understand it. They are activated. Now these nucleotides, they are getting activated by the ATP. ATP is the energy in presence of the enzyme phosphorylase. Now see, what is changing the nucleotides? These are the ATP molecules. ATP molecules, these are the energy molecules. Okay, and phosphorylase is the enzyme. This results in the formation of deoxyribonucleotides triphosphates. That means when there is phosphorylation, the process is phosphorylation. In the phosphorylation process, there is addition of phosphate group. Okay, so phosphorylase is the enzyme, and addition of phosphate group, that process is known as phosphorylation. Okay, so what by with the help of these two, that is in the presence of enzyme phosphorylase and the process that is called as phosphorylation, what is the result that we are getting in this step? That the nucleotides which are in the form of monophosphates, they are getting phosphorylated into another nucleotide. Their form is different, and from the inactive form they are getting activated. That is very important. That is, after activation, these monophosphates, they are converted into ATP. Now this AMP monophosphate, it gets converted into triphosphate. So there is addition of phosphate group. Try to understand that. Okay, GMP, that is guanosine monophosphate. Now see, monophosphate, mono means single phosphate compound, and when you are talking about GTP, that means there is addition of phosphate group, that means guanosine triphosphate. Another is cytosine monophosphate is getting converted into CTP, that is cytosine triphosphate. TMP, thiamine monophosphate is getting converted into thiamine triphosphate. Okay, and this process is called as phosphorylation. The second step in the DNA replication is the point of origin. So DNA replication begins at a certain or a specific point, okay, at a certain specific site situated on the molecule, on the double-stranded helical structure of the DNA, or which is twisted, which is supercoiled. We have studied the DNA structure. So on that double-stranded DNA, there is a special origin, a specific point. That point is termed as initiation point, from where actual the replication process it begins. Okay, initiate means to start, to initiate, to begin. Okay, so that such sites which are present on the DNA replication, they are called as origin points, or they are called as initiation points. In the eukaryotes, there are several origin points which are present on the double-stranded DNA structure of eukaryotic DNA replication, if you are talking about. But during replication of the DNA molecule, so that DNA molecule is split by the activity of enzyme at the origin by formation of incision fork or nick. Nick or incision means to cut. Okay, it initiates breaking of hydrogen bonds between the two strands, which is called as unzipping. So here in this slide, we are not talking about the eukaryotic replication. As you can observe this diagram now, this is related to the prokaryotic replication. This is a circular DNA of prokaryotes, and now you can see, see this circle, one circle is getting replicated or getting copied into another. Okay, so there is a single initiation point if you are talking about the prokaryotic cell. So as the replication process starts or initiates at this point, so now observe this diagram. At this site, the replication starts. Okay, so that point is called as replicon. Replicon is the unit where the replication occurs. So prokaryotes, they have only one replicon. Okay, so that is shown in this diagram. As we have seen in the previous slide, that in case of prokaryotic cell or prokaryotic DNA, there is a single replicon or a single origin or a single initiation point. Okay, but while talking about the eukaryotic cells, the situation or the condition is different. They have multiple origins. Eukaryotes, they have several replicons, more than one. The origin points, they are present on the eukaryotic DNA. At the point of O, the enzyme endonuclease cut one of the strands of the DNA. You can observe in this diagram, see, this is origin of replication. Now this is a double-stranded DNA, and this is origin of replication. This is single one origin, this is another origin, and this is third origin. Okay, now see, the bubble is getting formed. There is a replication fork, 1 2 3. So at the same time, there are 3 replication forks which are working, okay, or they are in action. And after this replication fork, see, this two daughter DNA molecules, they get formed. So this nick or that cut occurs on the sugar phosphate backbone or phosphodiester bond. So we have studied that. Now see, this is double stranded DNA, and these yellow colored dots, these 1 2 and 3 yellow colored dots, these are the 3 replicons or 3 origins which are situated on the double stranded DNA. And now see, these are 1 2 and 3 bubble formations. These are blue color is the parental DNA, and another red or red colored DNA which is forming the replication fork. Now this is newly formed DNA. Now you can observe that there are two colors. So this is one old strand and another is new strand. Again, newly formed DNA and old strand. Okay, so the point is, in the eukaryotic cell and in the prokaryotic cell, the replication origins or the initiation points, they vary. In the prokaryotic cell, there is only one cut, one incision, or one origin. That is a single point which is present on the circular DNA of the prokaryotic cells. While talking about the eukaryotic cells, the slide which is on your screen, so in the eukaryotic cells there are several replicons. So more than one number of initiation points are present because of the activity of enzyme, that is endonuclease. Now this name of the enzyme is very important if you are writing this second step, which is given in your book. Okay, so that's very important because that endonuclease, endo means inside of the DNA, endo means inside, exo means outside. Okay, so from inside, nuclease means to cut, to dissolve, or to cut. Okay, so when the double-stranded DNA, it gets cut from inside, so because of that there is formation of origin. Because of that incision, the origin get started. Okay, so the name of the enzyme is very important, and where the nick is getting occurred, that is on the phosphodiester bond. So that's I have explained in very much detail in the earlier lesson when we have studied the DNA structure. The third step, which is a part and which is very important when you are trying to understand the replication of DNA, is the unwinding of the DNA. As you can understand, the DNA double helix is completely coiled and supercoiled structure. Okay, so there is requirement when the cut occurs in the previous step, the enzyme endonuclease, it cuts the double-stranded DNA to the phosphodiester bond. So there is cut, but after that, the unwinding of DNA is very much important. When the DNA double strand is twisted, so that untwisting is very important. Okay, so there is one another enzyme which is playing a very important role in unwinding of the DNA, and that enzyme is helicase. So helicases are the enzymes that bind and may even remodel the nucleic acid or nucleic acid protein complexes. There are DNA and RNA helicases. So there are two different kinds of helicases. One is DNA helicases and another group is RNA helicases. So DNA helicases, they are essential during DNA replication process because they separate the double-stranded DNA into single-stranded, allowing each strand to get copied very correctly without any mistake. So that is very important. So the enzyme you can observe in the diagram. Now see, this is the double helical DNA strand which is completely twisted. Okay, now this is helicase which unwinds and separates the DNA strand. That is very important. So the enzyme called helicase breaks the weak hydrogen bond. Now these are the weak hydrogen bonds which are present in between the two strands of the DNA. So there is separation. Beautifully the helicase, it moves forward and it breaks and separates these hydrogen bonds. Okay, and it separates the two strands of the DNA. The two strands of the DNA get separated and they unwind. This unwinding is bidirectional. You have seen in the previous eukaryotic replication that there is bubble like structure. So when we are talking about bubble like structure, so at this position the DNA replication fork, so at this point this helicase is working. At the same time there is unwinding of the DNA at this point 2. So there are two types of helicases which are working together at this point, where the helicase is moving, and this there will be unwinding of DNA at this direction. Okay, and at the same time, this bubble, if you are talking about at this position, again another simultaneously parallelly another helicase will move forward, and that point, at this point, from that point, the DNA double helical structure will unwind it. Okay, so there is bidirectional. This is a one direction and this is another direction. So this is bubble formation, which we have studied in the earlier slide. Okay, so by this unwinding or bidirectional process, there, as it is continuous process, there is formation of Y-shaped replication fork. We are going to discuss this Y-shaped replication fork, how it gets formed. So each separated strand, it acts as a template. So see, now this helicase, it moves forward, and these are two different templates now. There is one DNA template from which a new DNA strand can get copied, can get formed, and this is another template. So they act as template. They act as old strands on which the newly synthesized DNA can get formed. That's all for today's session. Thank you.